So now that we're familiar with the basic structures of the neuron as well as the neuron types, let's look at the physiology of how neurons create and transmit signals. So here we're going to consider how an individual neuron responds to a stimulus. So first, this stimulus will create what's called a graded potential in the dendrites of the neuron, because remember that the dendrites are the receptive regions of the neuron, and the dendrites function to detect stimuli. So this graded potential is a short distance electrical signal that spreads from the site of the stimulus towards the neuron cell body. And it also even spreads, if it's strong enough, it will spread to the axon hillock, right? This cone-shaped region where the axon meets the neuron soma here. So if this graded potential is sufficiently strong, when it reaches the axon hillock will create an action potential. And that action potential will then spread like a wave down the length of the axon. And upon arriving at the axon terminals, it will cause the release of neurotransmitter. So those are the basic things that are happening when a neuron is stimulated. We'll look at each of these in more detail in the next few lecture segments here. But that's kind of the general idea of, or the general sequence of events for when a neuron is stimulated. So what types of stimuli are there? We talk about stimuli, but we haven't really talked about the types of stimuli that neurons are responsive to. So on the left here are the chemical stimuli, and then other stimuli types are on the right here. The chemical stimuli include things like neurotransmitters, odors, and taste. These are just chemicals that are dissolved, or that are aerosolized, that is, they're moving through the air, or chemical stimuli like taste are chemicals that are dissolved in our saliva and are detected by our tongue. We also have a number of other chemical signals that are present throughout the body that sometimes we're aware of, sometimes we're not aware of. Damaged tissues will release certain types of chemical stimuli that will facilitate inflammatory responses and immune system responses, so that damaged or dying tissue can be dealt with. We also have other chemical signals like hormones and things like that. And so those are the types of chemical stimuli that we'll mainly encounter, at least in our discussions here. Other types of stimuli, things like temperature, mechanical stimuli, which includes touch, pressure, and stretch in our skin, our skeletal muscles, and our joints, and things like that. So we see a number of mechanical stimuli associated with the integumentary and the skeletal systems. We also have light as a stimulus. So light is primarily detected by our eyes, and we have special sensory receptors in there. We have special neurons that are there to detect light and form images so that we can see. And so those are the other types of stimuli. So our focus here is going to be what happens when a neurotransmitter stimulates a neuron. So the type of stimulus that we'll be discussing in our lectures, or as we explore the physiology of the nervous system, is we're going to assume that a neurotransmitter is stimulating a neuron. We're going to learn a little more detail about these other types of stimuli in chapter 13 when we look at sensory receptors in the peripheral nervous system. So given that our focus is going to be on neurotransmitter signaling, we're going to look at signaling between two neurons here. So in this example, this cell releasing the neurotransmitter up here is going to be referred to as a presynaptic cell, or a presynaptic neuron. So anytime you have a neuron that's releasing neurotransmitter and signaling another cell, we're going to refer to that neuron that's releasing the neurotransmitter as the presynaptic cell, because it is the cell before the synapse, or before the synaptic cleft, because presynaptic just means before the synapse. The postsynaptic cell is the cell that's after the synapse, and this is the cell that's going to be responding to the neurotransmitter that's released. So our focus when we're looking at neurotransmitter stimuli is we're going to look at what happens in the postsynaptic cell when neurotransmitter stimulates this particular neuron here. So in order to understand how the voltage changes during graded potentials and action potentials, because remember that graded potentials and action potentials are voltage changes that occur when the neuron is stimulated. So in order to understand how these voltage changes occur during graded potentials and action potentials, we really need to understand the voltage difference across the plasma membrane when the cell is at rest, that is, when it is unstimulated. So let's briefly review resting membrane potential. So this is the membrane voltage that is seen in pretty much all of our cells, neurons in particular. It's very important. So this is the resting membrane potential or voltage that we see across the plasma membrane when the cell is not stimulated. So this is a good diagram, I think, that summarizes kind of everything that's going on in the resting potential or resting membrane potential or voltage of a neuron. And recall that resting membrane potential is a voltage difference across the plasma membrane. So it says resting membrane potential, but you can trade out this word potential for voltage. So resting membrane potential or resting membrane voltage mean the same thing. So if we were to put a small voltage meter or voltage probe across the plasma membrane, and if we read the difference in voltage between the outside of the cell, the extracellular fluid, and the inside of the cell, the intracellular fluid or the cytosol here, we would see that the cell has a slightly negative voltage on the inside. And the reason for this voltage is that potassium, which is a positively charged ion, it's shown here in orange, is constantly diffusing out of the cell. And because potassium is a positively charged substance, when it moves out of the cell, it creates a negative voltage reading, which is indicating, this negative voltage reading is indicating that there's positive charge that's moving from the inside of the cell to the outside of the cell. And so that's why we get the negative voltage that we see. So at resting membrane potential or resting membrane voltage, we see that there is a higher concentration of potassium on the inside of the cell than there is on the outside. And so that's what this diagram is showing here. So we've got a lot of potassium in our cytoplasm, cytosol, intracellular fluid, whatever you want to refer to it as, we've got a lot of potassium in it, right? And so that means that because of what we know about diffusion, potassium will diffuse from an area of high concentration to an area of low concentration. So that means that at rest, potassium wants to leave the cell. The opposite is true for sodium. So we see that at rest or at resting membrane potential, there is a lot more sodium, which are the orange spheres here there's a lot more higher sodium concentration on the outside of our cell in the extracellular fluid than there is in the cytoplasm or intracellular fluid at rest. So it turns out that potassium leaks out and sodium leaks in during resting membrane voltage, which is what we had just discussed previously. So potassium diffuses out of the cell through really abundant potassium leak channels, and so that's what we're seeing here. So these are just facilitated diffusion, which means that potassium is diffusing from areas of high concentration inside of the cell to areas of lower concentration outside of the cell. So at rest, potassium wants to diffuse out. Potassium easily diffuses out of the cell because there are many of these potassium leak channels. That is, there are a high abundance of these in the plasma membrane. And so this is what it is meant when the graphic here says the membrane is highly permeable to potassium, because there are a lot of these potassium leak channels that allow potassium to diffuse out of the cell. So sodium, which is also a positively charged ion, diffuses into the cell through these less abundant sodium leak channels. And the fact that there are fewer sodium leak channels in the plasma membrane means that sodium does diffuse in, but it's not as easy for it to diffuse in because there's not as many of these sodium leak channels in the plasma membrane. So for every one of these sodium leak channels, there are 25 potassium leak channels. So So when they say that the membrane is highly permeable to potassium, that means that, or that implies that there are a lot of potassium leak channels, and we see that that is the case. There are for every one sodium leak channel, there's 25 potassium leak channels. So there's a good abundance of those potassium leak channels in the plasma membranes of things like our neuron. So if only potassium was leaking out of our cell, and say we had no sodium leak channels, our resting membrane voltage would be minus 90 millivolts, which is a little bit more negative than it typically is, because only potassium is leaking out of the cell. But because sodium is leaking in and it's bringing some positive charge in with it, it's going to make the voltage a little more positive, or a little less negative, if you want to think of it that way. And so we end up with a resting membrane voltage of minus 70 millivolts there. So if only potassium was leaking out, our resting membrane voltage would be minus 90 millivolts. So it would be about 20 millivolts lower. So with potassium leaking out and sodium leaking in, the sodium-potassium pumps are necessary to maintain the concentration gradients of sodium and potassium across the membrane. So the book uses the analogy of this leaky boat here. So it describes the sodium-potassium pumps as maintaining the concentration gradients of both sodium and potassium by kicking some of that sodium that comes into the cell out, and bringing back in some of the potassium that has leaked out, and bringing it back into the cell interior. So a cell at rest is essentially like a leaky boat, with potassium leaking out of the potassium leak channels, sodium leaking in through the sodium leak channels. And so it compares the sodium-potassium pump to a pump that bails out excess water in the bottom of a boat to keep the boat floating. And so they use this analogy of the bailing pump for the boat. In this case, the boat is the neuron, which is transporting some of that sodium that's leaked in back out to the extracellular fluid, and some of that potassium that has leaked out, it's going to bring back in through the sodium-potassium pump. It's going to bring it back into our intracellular fluid. And so that kind of wraps up our review of resting membrane potential.